All right, hi everyone. For lab this week, we are going to be measuring xylem vessels. This is a common trait that you'll see in a lot of plant physiological work. The diameter of a xylem vessel can tell us a lot of information about water use efficiency, and also growth abnormalities in the plant as well. So to get a little practice doing this, you're also going to develop some skills in using a image analysis software that's freely available called ImageJ. ImageJ has been used by a lot of different types of science. And basically, if you need to measure things or count things or whatever sort of application you might need from an image, ImageJ can do a lot of stuff and it's free, which is really nice. So I am going to share my screen and walk you through the process of downloading ImageJ, setting the scale to make each image standardized, and actually measuring xylem vessels and keeping track of that data. All right, so hopefully you are able to see my screen at this point. We are going to begin by downloading ImageJ. So to do that, you can follow the link that I directly sent you in today's handout. Otherwise, just go to ImageJ and go to the downloads. They do have an online web portal that you can use just online without having to download anything, but I find that it's not nearly as good as using the downloaded version. So go to download, and then you would select, whether you have a Mac or a Windows. You download this package to your own computer. You'll find that it downloads in a file with a bunch of other files together here in ImageJ. So wherever you tell it to download, find it there. And you need to keep the application, which is this application icon here with the little microscope. This needs to be in the same file as all of these other plugins and macros and other files. So you can't just drag this out onto your desktop and keep it there by itself. Feel free to make a shortcut to it, but the application does need to be kept in the same file as everything else. When you open ImageJ, you will see this pop up on your screen. It's this is your toolbox. So if you've gotten to this point, you've successfully downloaded and opened the application. All right, so our first step here is to open an image. So you would go File, Open. You can also just do Control-O, and you would open your photo. And in this case, I'm going to look in my downloads for oh, I want desktop. My bad. All right. This is just an example image. So this is tree number 11. And we previously took fresh cross sections using a razor and getting a very thin section. These were mounted on a slide and magnified times 10. And what you'll see here is a little indicator arrow that was automatically in our field of vision through the microscope. If you look in here, you can actually see the xylem. So you'll see that the xylem vessels, which are each kind of circular little thing right here, are oriented in rays going from the pith outwards. And on the inside, there's phloem. So we don't want to get confused with that. But these bigger kind of circular areas are the actual xylem vessels. And each cross-section image, you might be able to see a few different xylem bundles. Some are going to be better fields of view than others, just based on how the microscope was positioned. So I would encourage you to select whichever one is the easiest to view and has the clearest, cleanest imagery of those xylem bundles. Okay, so this is what we're looking at. It's just a small snapshot of the total plant and all of the xylem that would be in the plant. But for this exercise, we will be measuring as many xylem vessels, which is an individual circle, as possible within one of these bundles. So don't measure them for everything, just choose the most clear bundle and measure all the xylem vessels within that. But before we can do that, we need to set the scale. I have taken the liberty of already figuring out what the true diameter of this indicator arrow is through a series of other steps. But so I've already measured that the width or the diameter of this indicator arrow is 0.044 millimeters. So to standardize measurements throughout all these photos, we're going to use this indicator arrow as our standard measurement. So what I'd recommend you do is use the Control+ buttons to zoom in on this arrow. And then we're going to select the line tool here and draw a line directly across that arrow for its diameter. Okay, you should see a little yellow line pop up there. Then you're going to go to Analyze, Set Scale, and now you can adjust it. So what the ImageJ program is doing is measuring that distance in pixels, but now you're going to tell it that that number of pixels corresponds with a known distance. So you'll adjust the known distance to 1.00 and the unit of length to millimeters, and then press OK. Now, when you draw a line that's ideally the same distance, right, roughly, you can see, you know, it's automatically calculating at the bottom of the toolbox there. You can see length on the far right, it says 1.08. Well, that's pretty close. We're somewhere in there. So you've correctly set the scale now. All right. So let's go back. Let's zoom out again and now zoom in towards our clearest xylem. And again, you'll see, you know, quite a few of them here, different rays, and we want to measure each one. And you also want to keep track of that data. So now what we do is use the line tool to measure the diameter of the xylem lumen. And what that means is the interior, this kind of open space, not the, you know, cell wall around it, but just the from inside to inside wall of that xylem. While your line is still fixed, click Analyze and then Measure. And ImageJ should now pop up with a table that shows you the number of whatever that measurement was. So this is your first measurement, it shows as 1. And then the column we're most interested in here is length. So that's 0.526, which doesn't seem right. Let me make sure. JK, that is correct. It's about half of the diameter. That does look okay. All right, so again, we want to measure as many xylem vessels as we can. Feel free to zoom in more if that helps. Sometimes the image gets a little pixelated as you go in, but if you can still see the circles pretty well, go ahead and zoom in. So we'll continue to measure the xylem diameter, and I recommend that you do it in one direction. A lot of these xylem are kind of ellipsoid or oval shaped, or they might be bigger in one direction than the other. I would kind of orient yourself with maybe the maximum diameter and measure all of them in the same direction. So again, draw your line, click Analyze, Measure. Analyze, measure. Analyze, measure. Now you want to come up with a system for yourself so that you're not accidentally measuring the same one over and over again. So I like to start from the top outwards and move your way in on a single ray. And just continue that way. Analyze, measure. And so on and so forth. If you're unsure about whether something is really a xylem vessel or not, you can note that you're unsure about it. Otherwise, just avoid it and try to get as many measured as you can. Okay, so let's say that you have finished all of your xylem here. You're feeling confident about. You should come over here to your total data set, and you should have maybe between 15 and 20 different xylem vessels measured. All right, so you would, I'm not sure if you can just highlight one column or not. But let's say you get everything and bring it over to a Excel file. And here's a little trick. If you want something transposed in the opposite direction, right now we have a vertical stack of this column. If we copy it and do a special paste special, we can transpose it and then it goes from left to right. So that might save you some time dragging things around. But you should orient these individual xylem measurements on this data sheet as follows. So your data sheet should have a column for the plant ID, and that plant ID is indicated on each image, right? This is plant number 11. All right, and then you should have columns for every vessel ID. So 1, 2, 3, 4. Let's say you can label it up to 20. If for some reason you have fewer than 20, then you would write NA in these columns. And then your measurements should then be placed in the proper row for whatever measurement you're taking. I already did number 11, so they're all here. Now, you should also have a column for mean vessel diameter in millimeters. And you can figure that by using an equation in Excel. So if you use the equal sign and then average, open parentheses, and then highlight all of the cells you want it to calculate that average from, and then close the parentheses, it will automatically figure your average for you. All right, and after you finish the assignment, keep this data sheet and turn that in on Canvas.